Polyketides natively have an unfortunate tendency to exist in organisms that are difficult to bioengineer. Many of them are fungi or bacteria that simply aren't genetically tractable. The difficulty of installing a DNA into a cell is what primarily defines how many genetic constructs you can test in an experiment. Because of this fundamental bottleneck, it is far easier to engineer a pathway once you have moved it into a genetically easy organism like E. coli or yeast. In this study, Khosla and co-workers moved the biosynthetic pathway for a macrocyclic core of the polyketide antibiotic erythromycin into E. coli. There are several problems that were encountered in this study and have since been encountered generally when trying to move PKS or NRPS pathways into E. coli. First, the polypeptides are unusually large for E. coli. The largest E. coli proteins are around 1,200 amino acids, and megasynthases are often 2,000 amino acids. Though many of the organisms that natively produce these enzymes are prokaryotes, the translational and chaperone machinery is different enough between these organisms that these genes often do not express well in enterobacteria. It is common to observe low protein abundance or non-functional misfolded proteins. Second, the megasynthases need to be pantethenolated to supply the thiol linker on each ACP domain. Though this all boils down to adding another enzyme to add that group, sometimes an appropriate enzyme is unknown. Sometimes one of the monomer units is not produced in the chassis. In this biosynthesis, propional-CoA is going to be the initiating unit. This metabolite is made in E. coli, but in low abundance in flux. The methylmalonyl-CoA extender unit isn't made in E. coli at all, so pathways would need to be added to provide these. Finally, E. coli doesn't naturally produce anything from propional-CoA or methylmalonyl-CoA, so it's unlikely that the cell's primary metabolism is optimum for their production. Flux through these precursors would likely need to be optimized by the usual means of promoter switches and genome knockouts. To get the protein expressed and properly pantethenolated, they note that pantethine is already present in E. coli. It is a component of coenzyme A. However, when they express the DEBS gene from deoxyerythronoline in E. coli, the enzyme doesn't get modified. To resolve this, they add the SFP gene from B. subtilis. In that host, this enzyme transfers panthenyl groups to the megasynthase for production of surfactant. Though surfactant and deoxyerythronolide have no particular structural similarity to one another, they are similar enough from the perspective of the panthenyl transferase. However, not all of these transferases have the same specificity, and they don't all work on all PKS modules. There were also difficulties expressing the polypeptides. All three DEBS genes produce protein, but only DEBS1 was functional after purification in vitro. They found that by lowering the growth temperature to 22 degrees Celsius, they could get the other two to express as well. This trick of reducing the temperature is often done in producing protein for crystallography. It helps proteins fold rather than aggregate by slowing everything down. They need to produce large amounts of propional-CoA and methylmalonyl-CoA, so they knock out the PRP operon encoding propionate catabolic genes to avoid degradation of this key intermediate. They also overexpress E. coli PRPE, which ligates propionate onto CoA to increase flux through propional-CoA. This intermediate, propional-CoA, is also the penultimate step to methylmalonyl-CoA. They add the propional-CoA carboxylase genes PCCA and PCCB from S-celicolor to catalyze this reaction. Note that this enzyme is fixing carbonate, which is just carbon dioxide dissolved in water, by incorporating it into the substrate as a carboxyl group. This type of chemistry requires the cofactor biotin and ATP. The biotin is a chemical modification to the PCCA subunit of the carboxylase. It turns out they need to overexpress the E. coli BIR-A gene encoding biotin ligase to get PCCA well biotinylated. Thus, the total modifications needed to observe product formation was a substantial effort. I find this paper particularly interesting because the things they must do to get the system up and working have all very reasonable, logical, and explainable things. However, they also get a little weird. It is easy to think about a complicated genetic device in terms of a black box diagram and forget the biochemical details of what goes on inside. This paper clearly illustrates how a deeper understanding of the biochemistry of the system can be essential to getting a device up and running in the cell.